Hello, my name is Sam Shepard. I'm a researcher, spent 37 years in developing technologies for a number of fields, in a lot of industries worldwide. What I'm about to talk about is primarily the effects of astaxanthin on cancer cells and some of the research that we've been able to develop and compile showing the recent studies on astaxanthin's effect on PDL1 binding to tumor cells. The two primary questions that always are asked is what causes cancer and how can I reduce my risk of getting cancer. I'm going to deal with both of those issues here, but first I want to talk about a normal cell. A normal cell typically is alkaline. Anything above a 7 pH is considered alkaline. Anything below 7 pH is considered acidic. Typical cells are about 7.35 to 7.4 pH. Something very important to watch in your blood work. The electric action potential, which is the voltage drop, and I'll discuss this a little more, between the inside of the cell and the outside of the cell, is minus 70 millivolts. The reason it's minus because the inside of the cell is slightly more negative than the outside of the cell. But it has a voltage across that membrane. A normal cell has express proteins. These are for normal function of the cell. This is a way that one cell will communicate with another. And the cellular metabolism, the reason we breathe in air and oxygen, is because these cells require oxygen. And they're known as aerobic cells. In a normal healthy cell, you can still see the negative 70 millivolt voltage potential across the cell membrane. The outside of the cell, high sodium levels, very little potassium. Inside the cell, it's the opposite. We have high potassium levels, very little sodium. This is a normal cell. We have also negatively charged proteins on the inside of the cell, which makes the inside of the cell slightly more negative than the outside. These are known as gate channels, sodium channels, or potassium channels. These are voltage regulated in most cases, and they will allow, depending on the voltage across that cell, whether to throw sodium out, let potassium in, or to bring sodium in and throw potassium and calcium and zinc out. So what causes cancer to form? Our research and studies so far, and there's a report that's issued, and you can contact me and I'll send you that report. Most cancer cells have high sodium levels on the inside of the cell, completely opposite from normal cells. They have higher potassium levels on the outside. This is a pretty serious issue. And one of the things that we found, one of the ways that we can convert a normal cell into a cancer cell is simply by depriving it of oxygen and raise the sodium level. At that point, there's a very high percentage of normal cells that will convert into a cancerous or anaerobic cell metabolism. So what does sodium do? Sodium levels cause action potentials across the cell membrane that changes those. For example, in a normal cell, we're minus 70 millivolts. In a cancerous cell, depending on the aggressiveness of the cancer, it varies from about minus 18 millivolts to a plus 33. Sodium levels will buffer the pH in a normal cell. What that means is that sodium chloride itself has a 7 pH. But if it's in an alkaline environment and the sodium level begins to increase, that will have a tendency to depress that 7.35 pH down to 7, more towards the acidic side. If you have an acidic cell, say a 6.2, sodium and sodium chloride will tend to raise it back to 7. So salt can have a tremendous effect on the pH of your operating cells. One other thing that sodium does, causes water retention. We all know the more salt we eat, the more we retain water. In a tumor cell, since the inside of the tumor cell has been shown to be very high concentrations of sodium relative to a normal cell, that sodium attracts water, causes the tumor cell volume to increase dramatically. The other more important thing that we found from sodium uptake, high sodium levels increases in what's called salinity between the capillary that's bringing the oxygen to the cell and the cell membrane is filled with interstitial water or fluid. Once that salinity of that fluid increases, the ability of oxygen to transport from the red blood cell to the cell membrane is greatly diminished. That concentration gradient no longer drives oxygen into the cell. For example, fresh water has about 14 parts per million of oxygen solubility. Salt water has 4. So the concentration gradient, or the driving force to drive oxygen from our red blood cells to the cell for metabolism, is greatly diminished because of the amount of salt that's between those cells. With that, the level of oxygenation that we measure in our bloodstream may be perfectly fine. That's not the problem. The problem is getting it from the capillary directly to the cell. Once that cell begins to experience a lack of oxygen, it goes from operating aerobically to operating anaerobically. All cancer cells are anaerobic, and the primary food that an anaerobic cell will need is sugar. And that's why it draws in and tries to create more blood vessels coming to the tumor cell, is because it needs nutrients. The other very interesting part of our research is in tumor cells. We see the tumor cell is expressing ligands. These ligands are referenced here as PDL1 and PDL2. There's several of those that can be expressed by a tumor cell. If a PDL1 or PDL2 attaches to the PD1 of a T cell, which is our immune attack cells, it totally inhibits the T cell and turns it off. This is why cancer hides from our immune system. So if there was a way of binding to that end of that PDL1 or the PDL2 and stop this connection here, that T cell will go active, send out chemicals, and destroy that tumor cell. Here's a list of cancers that we found that are what's called PDL1 expressors. You can see the relative positive effects of PDL1. These are some fairly aggressive cancers. The higher this number, the more aggressive the cancer appears to be. And these are target areas. If we can find a molecule that will stick to those active reactive sites on the PDL1 so that we can block it. And what I'm going to go through now is a molecule, a natural inhibitor called astaxanthin. This was the molecule that we did most of our research, and then a lot of research has been completed with this. And you can see the chemical structure. It looks like a dumbbell. You have a big head here and a big head here, but they have a double bond oxygen and a hydroxyl group. Hydroxyl group, double bond oxygen. This is what it looks like in a ball formation of the representation of the molecule itself. These are highly reactive groups. This is how astaxanthin appears to behave in a tumor cell. There's several different ways that it manages it. It's extremely reactive, a high ability. It's an antioxidant. It's an anti-inflammatory, because this is extremely reactive, and it has a tendency to hold a lot of electrons, and it balances out the electric charge potential across that membrane. So we have a head sticking outside. The length of this is almost the exact length of a typical human membrane, phospholipid membrane on a cell. Extremely reactive on the top, extremely reactive on the bottom. these molecules will tend to regulate the pH levels and the electric charge across that cell membrane. On the PDL1 and the PDL2 expressed proteins, these active sites on the PDL1 line up with these active sites of the astaxanthin. This acts as a blocking agent. So when a T cell comes in, tries to connect to that PDL1, it can't. It's blocked. Same with PDL2. It's blocked. So the T cell is not turned off by the tumor cell. So the T cell says, I'm going to kill it. It doesn't recognize that cancer cell as normal. And it says, I can now kill that cell. This is a very good example as to how it works. Here you have a T cell, and it's got a TCR and an MHC1. This MHC1 of the tumor cell has a small peptide. It's like a barcode. And this is coming in to read it to see if that peptide is of a normal cell or an abnormal cell. And in this case here, it's going to read it as an abnormal cell. If that PD1 of the T cell attaches to the PDL1 of the tumor cell, that cell becomes inactive and it will not attack that tumor cell. What happens with the blocking agents, for example, astaxanthin? The astaxanthin molecule, the reactive sites come down and attach to the carboxylic acids of the PDL1 and blocks it. This now cannot hook up to that PDL1. This will sniff that peptide. It will identify it as an abnormal. It will, the dendritic portions of the cell become active. Chemicals will be released, and it will dissolve that tumor cell. This is how chemically it looks. The astaxanthin molecule is attaching to the reactive site of the PDL1, the functional site. This is a carboxylic group here. That oxygen there will hydrogen bond to this. This being one bond distance greater than this, will have a tendency to wrap around and come around and completely block that functional group of PDL1. How does astaxanthin impart these? Reinstates the membrane electrical potential. That's very important for nutrient, fatty acid, sterol absorption. It's going to reduce the intracellular sodium concentrations because of the charge on either end of that molecule. It's going to increase the intracellular delivery of magnesium, potassium, and calcium and try to restore the cell back to normal. It's going to remove negative charges from the external surface of the cancer cells. The astaxanthin number 5 will inhibit the PDL1, PDL2 sites coming off of tumor cells. What this does, this is extraordinary, that it can actually stop the deactivation that tumor cells imparts on T cells simply by binding to PDL1, PDL2, and allowing the T cell now to remain active and to target and attack the tumor cell. The astaxanthin will correct the intracellular and extracellular and membrane measures of the reduced electrical action potential of cancer cells. It'll try to restore them back to normal, but what we've seen is that they really don't ever go back from an anaerobic to an aerobic cell. Typically, there's a possibility that it might happen, but typically those cells become identified as abnormal and they're either attacked or their cellular apoptosis sets in, which is cell death, or the cells are lysed and just broken apart. Number 7, reduces intracellular water accumulation. This is extremely important also. This is directly related to sodium intake within the cancer cell itself. So how do we reduce our risk of getting cancer from this study? 3 very important things to take into account. First of all, reduce or eliminate sodium intake by reducing extra dietary sources of salt. You still need sodium, you still need potassium. You get probably enough sodium and potassium in the food you eat, primarily through vegetables and cellular matter. So adding salt is a huge contributor to you acquiring cancer, strictly because of the hypoxic nature that sodium chloride imparts in converting an aerobic cell into an anaerobic cell. Reduce your salt content. The other thing is eliminate all sources of cellular hypoxia or oxygen starvation. Smoking is an obvious one. Smoking does two things. It not only creates carbon monoxide poisoning on your red blood cells, and it stops oxygen from actually getting to the cells, but it also has a negative effect on some of the protein structures and the pH changes that allows oxygen to, through a concentration gradient, to move from the red blood cell into the cellular membrane. This is really a double whammy. In spite of all of those things, those two things, it just so happens that when cells split in our body, you have one cell and it splits into two. If those cells are split into an area that is low oxygen demand, for whatever reason, strictly because of its location, that cell that's deprived of oxygen because of its location can still convert into a cancer cell through hypoxia. So it's very important that we don't have any cells that have that if they do begin to go anaerobic, we can use astaxanthin as the checkpoint block to attach it to the PDL1 and allow our T cells to take that cell out, either through cell lysis, and in most cases, the cell themselves will go into cellular apoptosis and destroy its cellular suicide. So those are very important. With that in mind, those three things: reduce salt intake, and drink water, drink plenty of water, because the fresh water will have a tendency to reduce the salt content also. But reduce your salt intake, stop any form of activity that may be causing cellular hypoxia, and take astaxanthin. If this is the case, you can contact me. We have astaxanthin available. It's a very concentrated form, but it is available. You can start taking it on a daily basis. This will probably reduce your probability in the 90th percentile from getting cancer. So feel free to contact me at any time. On the front part of this presentation was my telephone number and my email. I'd be more than happy to send you the report and also provide astaxanthin to you. With that, I want to thank you, and we'll go on to the next one.